Digestion and absorption, right? So far we've gotten things to move. We have added our secretions, and today we're going to do the work of the GI tract, right? So, we talked a long time ago about functional unit. What's the part of the system that does the job the body needs? Well, ultimately, absorption is the functional side of the GI tract. Everything else it does is for that purpose, right? Now, we need to have digestion happen in order for the molecules to be tiny enough and basic enough to be absorbed and be useful to the organism. Okay, one of the reasons that digestion takes big molecules and breaks them into small pieces is the nature of biology. We don't need cow protein. Cow proteins are not going to do us humans any good. But the amino acids that the cow proteins are made of, we can make our own proteins out of, right? That's pretty much the system. What nature makes are these big, elaborate, complex molecules. What we need for our own metabolism is small building block molecules, right? So we make that happen, that digestion happen by breaking things down. All right, so the most common kind of digestion chemistry, again, that we see is a hydrolysis. This should sound familiar, maybe not familiar because Gen Chem was a long time ago, right? But you did learn about this in Gen Chem, where you have a molecule that has two groups. You add a water to it, you get two molecules. One of the molecules has an OH, the other has the H, right? So stoichiometry is valid, but we've taken a large polymer, R double prime, R prime, and we've made it into a simpler molecule. Right? So that's hydrolysis, breaking it down. This system of equations is common across all of the food types. So carbohydrates, proteins, and fats all have a good deal of hydrolysis. The enzymes are different, right? Because the substrates are different, like you talked about in biochemistry a long time ago, right? Some reactions only can go forward when you have the right enzyme and the right substrate. So we have a lot of different enzymes, but the final outcome is the same, which is breaking the molecules into smaller pieces. Okay, so the idea is to go from monomer or from polymer to monomer. Now, typically, we can't do this in one step. Our digestive enzymes tend to go from macromolecule, intact protein, intact starch, intact fat, to a fragment. In other words, in a protein, you don't go from protein to individual amino acids. You go from protein to smaller proteins, and then those proteins get broken down into smaller yet, and eventually at the brush border, you finally get to your amino acids, right? So it's often a multi-step process. Same is true for starches. We go from starches typically to disaccharides, and then we go from disaccharides to monosaccharides, again at the brush border, okay? Now, I have split the thinking about digestion, absorption, and metabolism into kind of two pieces, and there's an exam in the middle, okay? So today we're going to talk about the basics, the concepts of digestion. What we'll start talking about after the break is what happens to those products in metabolism, right? So we're going to talk a little bit about amino acid metabolism, protein metabolism, carbohydrates, and fats after the break, okay? So this is really, today's talk is about how does the GI system accomplish digestion. After the break in metabolism and energetics, we'll talk about what happens next, all right? And you'll have post-traumatic stress from biochemistry days, because the Krebs cycle is in that one. The Krebs cycle, remember that one? You know the real Krebs cycle, right? You learn the Krebs cycle, you forget the Krebs cycle, and you learn the Krebs cycle. That's the real one, right? All right, but you'll be happy with the Krebs cycle when I'm done with you about it. Okay, so more than one step. Condensation is the opposite of hydrolysis. When we start talking about anabolism in our metabolic discussion after break or after the test, we're going to see a lot of condensations because ultimately that's how proteins are formed, right? It's a condensation